second half of our morning plenary session. It's my pleasure to introduce Dr. Rosemary Gillespie from the University of California, Berkeley, who will be talking to us about evolution of ecological communities through the lens of an island chronosequence. Thank you, Rosemary.
Thanks so much, Bob. And thanks to everyone for having me here. This is a tremendous pleasure, tremendous opportunity for me to talk about some of the work that we've been doing and hopefully trying to convince you of the value of a kind of model system for addressing issues in biodiversity science and for the use of the barcoding approach to do this. So just to start, I'm going to talk about a project, I'm going to focus on a project at least, that is a very large collaboration, NSF funded through their Dimensions of a Biodiversity program. And if I can get this to work. The two major players in the work that I'm going to talk about are Andy Rominger, who's a macroecologist, theoretical macroecologist, and Henrik Krehenwinkel, who's a genomicist. So what I want to talk about is really what the issues that people brought up this morning of biodiversity dynamics. We all want to see change over time, and we're limited in seeing change over time by our own lifetime. So when we look at biodiversity dynamics, the things we want to do are go up from microevolutionary processes where you're looking at lineages coming together and dividing and all sorts of things going on at the microevolutionary scale, and go up, scale up from that to macroevolution where things are actually diversifying. At the same time, we want to look at how evolution intersects with ecology. So evolution looks at exactly how things are connected over historical time, so how things are related, and you're always looking over the history. Whereas in ecology, you're basically looking at one point in time. You're looking at ecological metrics over space, but the time period is necessarily very limited. We're looking at very short temporal scales and looking at food webs and some really cool ecological approaches, but most of them are limited in terms of their temporal component. So what we want to do to understand biodiversity dynamics is put these two together and to use that to understand the actual dynamics of biodiversity. So what I want to try and see is how we can do this, because the processes occur over different scales, and so it makes it really hard to understand the dynamics. So what have people done? So just the ways people have used to get at this kind of problem is to integrate neontological and paleontological data. And this is just, these figures are based on a paper out of Catherine Graham's lab, where they looked at the dynamics of trait evolution. The blue lines are the extant lineages, the brown are where you include the extant and the extinct lineages. And the circles are the true and reconstructed trait values. And you see, when you include fossils, you get a much clearer picture of what the past what the lineage went through in the past. In the same way, when you look at species diversity through time, you can estimate species diversity through time based on extant lineages, but that gives you often a very biased view of what lineages actually exist, and you can get a better feel for that when you include fossil lineages. And likewise, when you look at the temporal dynamics, so niche interactions, we often use niche reconstruction based on extant lineages and go back in time based on what we know of current distributions. But often when we include fossils, we get a very different picture. So that's been a very powerful way, including fossils is recognized as tremendously important in understanding biodiversity dynamics. But there's another way to do this, and this is through the use of chronosequences. So this is what I'm going to focus on. an island chronosequence, and I'm going to focus on one particular island chronosequence because it's just a very beautiful one, and it behaves very well, and that's the chronosequence provided by the Hawaiian Islands. So what's nice about this is that in Hawaii you get a hotspot, which is just off the southeast coast of the big island of Hawaii. And so basically this hotspot oozes out lava, and as the plate moves away from the hotspot, the islands get older and eroded and eventually disappear. So you can use this chronosequence basically as a fossil record. So here you see 5 million years ago, Kauai was just forming. And these islands were low and far apart, and so they were actually, you can't do much with those older islands. Most things went extinct before they reached Kauai. So Kauai is kind of the first in the chronosequence of the current high islands. There are exceptions, I should say. So Kauai is the first, then Oahu came along. Things tended to jump from one island to the older island to the younger island. So the Maui Nui complex came along, jumped over there, and then you get Hawaii coming along, and they jumped over there. And so basically, you can use this almost as a fossil record, the oldest in blue, yellow, red, down to the youngest in turquoise. So you can basically read this as a chronosequence, as a fossil, read the chronosequence as a fossil record to look at the dynamics of trait evolution, species diversity through time, and temporal dynamics and niche interactions. So what I'm going to talk about then is microevolution, microevolution going to macroevolution. So evolutionary dynamics over time, species diversity through time, and ecological interactions over time. So first of all, let's look at microevolution to macroevolution, the very early stages in diversification. So for this, we're going to come to the youngest island of Hawaii. And I should apologize to Paul, much as he hates spiders. Much of this talk is going to be focused on spiders because that's what I'm most familiar with. But I should emphasize that all of the work we're doing is focused on arthropods as a whole. My particular focus is on spiders. So the work we've been doing to try and get at the microevolution to macroevolution stage focuses on the big island of Hawaii, where things are just starting to get going, and going over to the island of Maui, where you get clear, defined species. So I had a student, Amy Vandergast, who built on work that Hampton Carson suggested on this very young island. Basically, what you find is that the forest gets repeatedly carved out by lava. And as a result of this is a geological sequence on this young island. But if you look at this area here on this very young volcano of Mauna Loa, what you find is that it's very naturally fragmented by lava. And so you get these little green patches are islands of vegetation. And what Hamp Carson argued was that these little fragments serve as a crucible for evolution, basically carving things up and allowing them to come back together again. So what Amy Vandergast did for her PhD was look at how well these fragments were separated by lava. So in terms of the organisms that were restricted to these fragments, were they separated? Were they genetically disconnected between forest pockets? And what she found, this is just mitochondrial DNA and allozymes, what she found was when forest patches are separated by lava, indicated by the red, you find marked genetic differences over distance. So this is the distance. When they've just got continuous forest connecting them, they're just the same across that entire forest area. So the bottom line here is that forest fragments repeatedly are fragmenting these populations. So they're limiting gene flow. So what we're interested in now is this balance between isolation and time. So what happens is the lava comes down, separates taxa, and then they come back together. Over time, they come back together. And coming back together is necessarily going to either, it's not enough time for them to become new species, but almost certainly the fusion is going to inject diversity in the same way as it does for cichlid fish at the beginning of our radiation there as well. So what we're interested in doing now is figuring out how this dynamic, this fusion-fission cycle plays out over time to generate diversity and adaptive radiations. So going on then from this, we want now to look at evolutionary dynamics. So basically, this is what people have looked at when they've used the fossil record. They've said, okay, the extant taxa we can infer that this was their phenotype, this is their niche, but then when we include fossils, we get a different picture. So this is what they've done using fossil taxa. Now what can we do using the chronosequence? So let's just first of all think about what's going on with the chronosequence overall in Hawaii. So you've got the oldest island there, the youngest there. And there's a standard kind of dynamic that happens here in that most lineages progress from the oldest to the younger islands. Of course, there are exceptions, but this is a general rule. Now, let's just look at a few lineages as they hop down the island chain. This is one group, the genus Orson Welles, a sheepweb spider. And this is work by Gustavo Hormiga and Mikel Arnedo that they did a few years ago with me. And so what they found was that these species have jumped from the older to the younger islands. But let's look at how they've diversified. You see, on the youngest island, just one species. Then on the middle island, this group here, there's a whole bunch of species. On the older island, more, and just a whole pile of them on the oldest island of Kauai. So basically, this group has undergone very little adaptive radiation. They've simply changed in geographic space and become new species. And they've just accumulated over time, just more and more species. Now, if we look at another group, these are crab spiders and the work of Jessica Garb. What she found on the oldest island, you get most of the lineages on the oldest island within this radiation. And you get much of the ecological divergence on the oldest island. And then all they did was hop down from the older to the younger islands, keeping their ecological affinities. So here's a pattern of diversification early, adaptive diversification early in the radiation, and the populations just hop from one island to the next, maintaining their ecological affinities. So then come to just one more pattern that you find. This is the genus Tetragnatha that I've done a lot of work on, and this is a huge radiation of super cool spiders. And this is one group, the spiny-legged species that run around. They're active cursorial predators. How have they diversified? When you look at them, the oldest island going to the youngest, so they've hopped down from the older to the younger islands. But here, I've marked ecomorphs. These are very distinct ecological forms, the green ones, the brown ones, the maroon ones, the big brown ones, that have evolved repeatedly over this time frame of the Hawaiian Islands. So independent evolution of ecomorphs. You find exactly the same thing in these stick spiders, the genus Ariamnes. So what they've done, and you can't read this too well, but the point here is this is Kauai, where you get a brown and a gold ecomorph, Oahu, where you get independent gold, white, and brown, a whole bunch of them within the brown, gold, and white ecomorphs on Maui, and then just a couple of populations on Hawaii. So then how does this work in the context of a chronosequence? So just to explain, because sometimes it's hard to relate what we're trying to do with the fossil record with a chronosequence. But basically, here's what you're trying to do with the fossil record, get a more accurate picture of the deeper nodes. In a chronosequence, in an old chronosequence like Hawaii, we're looking at the oldest island, and here you see the extant representatives from that old lineage. But then it reset itself. It went to the next island, and reset itself. And so it started again. And here you get these 3 species based in that time frame. It went to the next youngest island, reset itself, and evolved all of these different taxa again. And then to the youngest island, where you just get populations appearing. So it gives you a sequence of change, basically a resetting of the lineage, so that you can see what happens in the early stages of the lineage and later and later and later. So what we can see based on this, then, is that taxa differ in the interplay between ecological and genetic shifts during diversification. So what we can now do is look at patterns of diversity over time across whole communities. So if you look at the taxa that I've just been talking about, this is the fossil record, and how the information from extant lineages in the blue is much clarified, and you get much more information by including the fossil record. Let's look at the chronosequence and see what we find. Here is the species per unit area of the spiders I've been talking about. The Orson Welles, the crab spiders, and these are the Ariamnes, the stick spiders, and the Tetragnatha. And you get very different patterns over the chronosequence. So some of them just increase just forever to the oldest island, the Orson Welles. Some very quickly reach a peak and then just stay steady. Some seem to reach a peak and die down. And these are the ones that undergo the repeated evolution of similar form. You find that across lots of different taxa. This is all sorts of different animal taxa in Hawaii. You get the more diverse lineages showing a peak of diversity on the middle-aged islands and then dying down. The smaller lineages keeping diversifying, keeping increasing. So relating this, the fossil, what they're doing with the fossil data, what you do with the chronosequence, what we're finding is different lineages show different patterns. Some just increase over time, some increase and then level off, and some show a peak and then die down a bit. So what we can say then is that taxa differ in the interplay between ecological and genetic shifts during diversification. We've seen that. But the rate of accumulation of species and the patterns of diversity over time are different for different groups of taxa. So this highlights that different groups perceive the chronosequence differently. And what we can do now then is look a bit in greater detail at ecological interactions. So we can actually use the chronosequence to give a temporal dimension to the explicitly spatial ecological theories that have been developed. So for example, here you see, this is where they've used the fossil record, what we talked about before, to look and see what distributions of the ancestral taxa in the fossil record were and how the current taxa relate to that. How then do we use the chronosequence to do that? So what we've done here is we're using this, a tool that, one that's been finessed really by John Hart, who's a collaborator in this work. And John uses an approach, the maximum entropy theory of ecology, not to be confused with the niche modeling use of this approach. But in this, what he's called MET approach, what he can do is just basically look at communities and develop metrics of abundance, diversity within those communities based on statistical properties of the communities. Basically, what it's doing is generating a null expectation. If no biology is going on, what kinds of distributions would you expect? In short, what it's basically doing is taking the energy of a community and packaging it most parsimoniously into individuals and then into species. So this allows us to look at community-wide patterns across the chronosequence and testing of this theory across the chronosequence. So here you see, based on very preliminary data, these are for detritivores, across the chronosequence. This is the oldest island, and this is the youngest. The black line indicates the predictions from the maximum entropy theory, the black across here, and the circles are the actual data. And what you can see is that you get the highest deviations from the max-ent predictions on the youngest lavas. So the deviation from the predictions on these youngest sites may indicate that the community hasn't reached steady state. All this is saying is that this is something to look at. It's not behaving normally as normal communities behave. Now, you can do the same with interaction networks to see how predictable change is over this time period. So what we've done so far is look at modularity and nestedness. We've developed just a bipartite network for leafhoppers and just generated these networks and tested them across the chronosequence, looking at how modularity and nestedness change across the island chronology. And what you see is that nestedness decreases with age, perhaps due to the high immigration of new species with high probabilities of eating or being eaten by general species already present. And then modularity increases, likely due to coevolution on the oldest island. Now, so this is just basic food web metrics. You can also apply the max-ent theory to these kinds of interactions. So using statistical theory to say whether you get statistical deviations from the expectation of food web interactions. Again, using the leafhoppers and the plants. And so here again, the red line here is the expectation based on the maximum entropy theory. The circles are the actual data. And what you see is that you get this is statistically not significant when the red line falls in the gray. And what you find is that they statistically deviate from the predictions on the very youngest island, probably because assembly hasn't really got going. And then there's another deviation on the oldest island, presumably because you get ecological specialization. So these are both indicating that networks deviate from statistical predictions based on these sites. So what we can say then at the moment is that across the chronosequence, different arthropod groups perceive, colonize, and diverge across the evolving landscape differently. And this results in different patterns of species accumulation over time. You get deviation from statistical steady state that varies across groups. And the interaction of ecological and evolutionary strategies, isolation and mixing, likely interact to produce the patterns that you see. So as we end here, what I want to just tell you a little bit about is what we're doing going forward using this chronosequence. So we've got a team out in Hawaii at the moment working across this chronosequence, the oldest island, and these are the actual sites that we've got. So we're looking at lavas of different age from the very oldest to the very youngest. I should say just here that we're looking at sites that are standardized as much as possible with similar elevation, precipitation. They're all metrosideros forest, and they're all accessible. What we do in those sites is we collect the bejesus out of arthropods. We're using very quantitative approaches to get actual numbers based on multiple sampling methods. We don't use lures or anything like that. We're just looking, we want to get numbers of arthropods at a given site. And so we use things like malaise, beating, litter samples, and multiple other techniques. So what we're wanting to do then with that data is to look at everything across the chronosequence. We're trying to get at biodiversity dynamics, and when you look at biodiversity dynamics over time, you want to get everything. So this is really what Henrik Krehenwinkel is pioneering here, looking at at metagenomics across the chronosequence. So what he's doing is trying to develop an approach that allows you to look at change in the evolutionary dynamics right across the chronosequence. So looking at the genetic structure of all the lineages in the ecosystem, and which factors drive divergence in lineages. So which ones separate first, and which ones follow after the others. Do bacteria diversify, and are they driving the process, or the fungi, or is it the interaction, or is it prey that start and then predators? What exactly is going on? And at the same time, what he's trying to do is get the data to give you actual abundances. So get rapid identification of taxa, because you get far too many, and look at relative abundance and use metabarcoding of whole predators to identify prey-predator interactions and food plots of herbivores, which will give us a much more precise indication of food-web interactions and how these are changing over time. So the multiple approaches that he's using, these are all in development at the moment, and I think you're all very familiar with the approaches that he's trying to pioneer. Basically, looking at everything, using all the tools available to him, and building on work of many others. So this is the work that's actively in progress, and I think it's a very exciting time to be using this kind of data to understand, to actually get a feel on how lineages change over extended time periods. And with that, I just want to say thank you, and I'd take any questions if there's time for any.